Yeah, I want to thank everyone, especially Chris and Britt, for having me, and especially for having me after this screw up last time I tried to do this. I was expecting something to go wrong, but it didn't, and here I am, so thanks. So I'm going to tell you about work that we've been doing over the last multiple years, and particularly done by these two terrific people that I've had the luck to work with, Dan Rubin over there, who was a grad student working with me, a MD PhD, who's now doing his neurology residency, and Yasser Ahmedian, a postdoc physicist who is about to start a faculty job at the University of Oregon. And we've also worked with Steve Van Hooser doing experiments to test the theory at Brandeis. Right, so the title of the talk is The Stabilized Superlinear Network, a Simple Circuit Motif Underlying Cortical Multi-Input Integration, or Explaining a Lot with a Little. And the basic outline is I'm going to tell you about the stabilized superlinear network, which is a little, just a simple idea about how a circuit works, and the biology it can explain, which I hope I will convince you is a lot. A moment to just talk about the philosophy of what are we doing by modeling, what are we trying to accomplish, and how do we approach modeling something as complicated as the brain. We take Einstein's dictum of our theory should be as simple as possible, but no simpler. So it's sort of a radically simplified picture. In this case, I'm not going to have any cell types other than just excitatory and inhibitory, ignoring all the recent work on the multiple inhibitory subtypes. Not going to consider spiking, just going to consider rate neurons, not going to consider extended dendrites and processing and complicated processing inside single neurons, just point neurons. Just going to consider processing in a single layer, which we think of as layer 2, 3, not dealing with the whole complexity of the multiple layers of cortex. But the idea is to understand basic dynamical motifs that can give a unified explanation of a range of phenomena, and that hopefully the basic ideas we uncover with these simple models will persist and be there underneath more complicated models with a lot more going on. So that the basic idea is that modeling is sort of a scaffolding. By studying a model, building it up, we arrive at new intuitions that we wouldn't have had without having done the work of modeling. New intuitions as to how circuits work, what their dynamics are. Once we have those intuitions, we don't necessarily need the scaffolding anymore. The intuitions now can help inform us. I mean, you still need the modeling to figure out exactly what you predict will happen, but you gain a new intuition that you can then use without the scaffolding that got you there, but you never would have gotten there just by thinking about it without really the formal modeling process. So what's the big picture, the big question that we're trying to address with this little model of a circuit? General problem is that your sense data arrives locally, and perception involves knitting these together into objects in a way that's invariant to, yet dependent on the details of the local data. So for example, if these little circles represent the receptive fields of different neurons in primary visual cortex, so what aspect of the visual world are they looking at, are they sensitive to? So a neuron here may be seeing both this oriented line and this curved line, has to figure out how to integrate those two things to determine its response. But in addition, there's all these other neurons in other points in V1 that are representing other pieces of this. And somehow all of these little aperture pictures need to be integrated together to give the idea that there's a glass here. And as we all know, it doesn't, I'm going to see a glass at all different poses, all different positions, all different lighting and shading and occlusion. So it doesn't depend on the exact details of the pixels, but something higher order that we're trying to extract. And this isn't just a sensory problem. You have the same problem and I should say the same thing applies to every sensory modality. Your touch, audition, they all come in locally in some sense, but you have to integrate a global picture. You have the same problem in the motor system. I can make a plan to point to that slide. I can point with my finger, I can point with this, I can point with my head. And so I have an overall plan which then gets implemented in some details and then get implemented in finer details, and yet the overall plan is invariant to the detailed implementation. So how does the cortex do this? We don't know. But we know a couple of things about the process by which it's done. One is that within any one area of cortex, well, there's a hierarchy of cortical areas, at least certainly we know that best in vision, starting from the areas, the primary cortex, primary sensory cortex that first receives the sensory information from the periphery, from the eyes, in the case of vision, and then a hierarchy of areas where within any one area, you have multi-input integration. So you have how does your neuron decide when it's getting two different orientations, how to respond to them. You have how does the neuron integrate the information in the surround to determine what its response should be. And then in addition, we have this hierarchical feed forward and feedback between areas where we see that sort of larger and more invariant object-like structures, or selectivity for larger invariant, more object-like structures emerges as you move up the hierarchy. So we know these two processes are involved in some way, that we don't really know how it works. Today I'm going to focus on this process, the multi-input integration within one area. So one piece of this general puzzle. So what do we know about multi-input integration within one area? Well, I'm going to give you here a simplified picture. I don't think it's too simplified, but I think the jury is out. But at least as a first cut, this seems to be how things are working. And that is that the feed-forward input, the input coming in from below, if you will, from the eyes, or by way of the thalamus to the primary visual cortex, the feed-forward sensory input coming from outside largely determines your selectivity. So, you know, between all these different orientations, which one do you respond best to, and how selective are you? How rapidly does your response fall off as the orientation of a line in your receptive field changes? That's pretty much determined by the set of feed-forward inputs that you receive. And there's an awful lot of work showing that, at least in primary visual cortex. Furthermore, if you're getting multiple feed-forward inputs, if there's a couple of stimuli in your receptive field, they mutually modulate one another's response gain. We'll go into this in more detail, but there's some process of normalization where you get a summation of the response to each individual stimulus, but it's a sublinear summation. It may be, for example, for two equal strength stimuli, it can be closer to an average of the two responses than a sum of the two responses. And so you can think of this as they're sort of mutually modulating one another's gain of response. In addition, you have contextual and surround, that is from the surrounding area, contextual modulation and top-down modulation of various forms, attention being the most obvious. And these also seem to primarily modulate response gain. They don't change your selectivity so much. And examples of this are surround suppression, where you're integrating inputs, and we'll talk more about this, integrating inputs outside the classical receptive field. The classical receptive field being the region of visual space for a visual neuron, the region of visual space where an appropriate light stimulus can drive you to spike. Outside of that region is the surround, where light stimuli there alone will not drive you to spike, but they can modulate the gain of your response to stimuli in your classical receptive field. And so that surround suppression is an example of that, or attentional modulation. And so what I'm going to be talking about today in particular is how do you integrate, how do these multiple feed-forward inputs mutually modulate one another's response gain, how do contextual and top-down modulation modulate gain. So a very simple motif that gives a mechanistic explanation of this dynamical in space, changing in space and time setting of gain. We'll be mostly talking about in space. And just the long-range goal of this, why do we want to understand mechanically exactly how the circuit does these operations? Basically because we don't know really what these operations are. We have characterizations of them like normalization, surround suppression that are based on laboratory studies of laboratory stimuli in restricted circumstances. We don't have a very good characterization of how the cortex processes real natural scenes in a real natural environment. And so if we can get at what's the mechanism, the hope, the dream at least, and the goal is to then understand what is the true underlying operation which manifests in these things we've identified in the laboratory circumstances. And if we understand that true underlying operation, then if we see how it processes natural scenes, we may begin to understand something more deep about what's the function, why is it processing things this way, how does that contribute to the overall object recognition. So that's the big picture, and we're going to be looking now at this particular picture, this particular issue. So outline of what I'll be talking about, I'll be telling you first about the basic idea of the stabilized superlinear network, and then we'll be talking about the biology it can explain. Normalization and stimulus-induced suppression of correlated variability, and surround suppression, which will be in several steps, which I separate out to help you keep oriented as we go through them. And that's the talk. So right now, we'll start by talking about the stabilized superlinear network. This is a simple, very generic mechanism that explains a transition from modulatory facilitation for weak inputs. You tend to enhance responses when inputs are weak, to modulatory suppression for stronger inputs. More generally, this gives a transition from a superlinear response summation. So you have a response to stimulus 1, you have a response to stimulus 2. When you see them both at the same time, you get more than the sum of their responses. That's what I mean by superlinear. Superlinear response summation for weak inputs, and a transition to sublinear response summation. I get less than the sum of two responses when the inputs are stronger. So it all starts with a property of single neurons in cortex, and that is that there is a superlinear input-output function where if we look, and this is a recording from Nick Priebe and David Furster in CAT V1, if we look, what they did was they looked at they're recording intracellularly in vivo and looking at the membrane potential in 30 millisecond bins and the corresponding firing rate in those same bins. And all the data is shown by all these gray squares. But if you average in voltage bins, you get the blue dots, which are very well fit by this red curve, which is a power law, in this case with a power of 2.79. And this is what they consistently find across neurons. And we know a theoretical reason for this, that basically cortical neurons are the mean input to a cortical neuron would not get it to threshold, or would only get it about the threshold. It's subthreshold, basically. And the neurons are firing on fluctuations in the input rather than on the mean input. When you're firing on fluctuations, then it essentially means that you imagine you have some Gaussian distribution of voltage fluctuations. And now as you move your mean, you're moving superlinearly more and more of that distribution above threshold. And the amount above threshold is basically your firing rate. And so that's why as you increase the input, your response rate is going up superlinearly. Now I'm telling you that from the input to the firing rate is a superlinear function. What I'm showing you is from the membrane potential to the firing rate is a superlinear function. That begs the question, well, what's the transformation from the input strength to the membrane potential? And that might be sublinear in certain ways. For example, the feed-forward synapses may be depressing, so that they're less strong when they're more active. And the postsynaptic cell may show spike rate adaptation, which will also tend to suppress the response when it's more active. Everything I'm going to tell you just hinges on this function being, overall function from input to firing rate being superlinear. And so I have to cross my fingers and make a wish and hope that however sublinear the transformation is from input to membrane potential, it's not enough to undo the overall superlinearity of the transformation. And I'll show you that if we do have this overall superlinearity of the transformation, this can explain an awful lot. It explains so much that it makes me believe that the real transformation has to be superlinear, but I can't prove that to you. Okay, so this is what they see. And so we're going to say that the steady state response to a given input current I is you rectify that input current. That is, if it's negative, I just get 0. And then I raise it to some power n. And what this does for you is that your gain of your postsynaptic response, that is, how much does my firing rate change for a given change in input, the gain is monotonically increasing with the overall level of activation of the neuron, because the gain is just the slope of this superlinear input-output function. And also, I want you to notice that in this example, and really across almost all the examples they look at, they do not see neuronal saturation throughout the cortical dynamic range. So we know ultimately the input-output function has to saturate. Neurons can't fire it at infinite rates. But the point is, over the whole dynamic range of visual stimuli that a neuron sees, from the most optimal stimulus you could give it on down, it never gets out of this superlinear regime. It never gets into it. It never finds the saturating regime. So somehow the network is keeping itself in this unsaturated regime. And part of what I'm going to tell you about is how it does that. And I should say, this is seen in CAT V1. There's a whole lot of work in rodents right now, and it really remains to be shown that this is going on in rodents. But we believe it probably is, but it remains to be shown. Okay, so growing gain with activation level means that you get growing effective synaptic strength. So what do I mean by an effective synaptic strength? It's basically when the presynaptic cell's rate jiggles, how much does the postsynaptic rate jiggle in response? So it's how much does this guy respond to this guy's fluctuation. And that's the product of two terms. There's the synaptic strength. When this one jiggles a certain amount, how much current does it inject? That's the synaptic strength. And then how much does the postsynaptic rate respond to that current? That's the gain. And so the effective synaptic strengths are growing as the gain grows. So now let's imagine some network of point rate neurons. So I've got some excitatory neurons. I've got some inhibitory neurons. I've got all the sets of weights between them. And then I've got whatever the feed-forward input is to these neurons, which might be different to different neurons. But the net vector of feed-forward input, what this one gets, what this one gets, what so forth. And so now when my feed-forward input is very weak, when I'm weakly activating the circuit, I've got weak effective strengths. And so the recurrent drive, so I've got the feed-forward drive and the recurrent drive. The recurrent drive is doubly weak, because basically the only way you get recurrent drive is that the feed-forward drive causes the cortical cell to change, which then changes the recurrent drive. So any term involving a recurrence involves two or more of these weak synapses, the feed-forward one plus at least one recurrent one. The feed-forward one only involves one weak synapse. So therefore, when the synapses are weak enough, the recurrence becomes negligibly small. You're dominated by the feed-forward input. And so basically, you're in a weakly coupled regime. You're close to uncoupled. And as a result, you're basically, each neuron is just independently seeing its feed-forward input. And since each neuron integrates its net input, feed-forward plus recurrent, integrates its net input superlinearly, and since it's really only seeing feed-forward input, the recurrence is tiny, then responses to different stimuli add superlinearly. Because you just add you get two different feed-forward inputs, you're going up that superlinear input-output function. So for weak activation, you're in this superlinearly summing regime. But with stronger activation, you have stronger effective strengths. That means the recurrent terms involving 2 and 3 and 4 synapses are getting bigger and bigger and bigger and ultimately come to dominate. So the recurrent contribution grows. And in particular, the recurrent contribution grows to the point where the E to E synapses get strong enough that the excitatory subnetwork by itself would become unstable. Basically, you can imagine if the excitatory subnetwork had some fluctuation of its rate, how big would the fluctuation of response to that fluctuation be? And if the fluctuation of response will be bigger than the fluctuation that started it, then your excitatory subnetwork has gone unstable. And that will happen once the gain is big enough. So you'll reach the point where the excitatory subnetwork is unstable, and then either your network explodes and goes epileptic, or it doesn't. And if it doesn't, it's because you're stabilized by feedback inhibition. And we can show sort of generically that if your feedback inhibition is strong enough, the network will find its way to this dynamic stabilization. But furthermore, something very interesting happens, which is that this is sort of a very explosive nonlinearity. That is, if you start to go unstable here, you go up to an even higher rate where your gain's even bigger. And so it's like a nuclear reaction going critical. It will just take off. It turns out that to stabilize against this explosive nonlinearity, the network has to, basically the recurrent input has to very largely cancel the feed-forward input, leaving a residual that grows slowly compared to the growth of the feed-forward input. So if I double the feed-forward input, the recurrence will cancel most of that, and maybe I'll get a square root of 2 or a cube root of 2 growth instead of a doubling of the net input. And as a result, the network response is basically the growth in the feed-forward, the growth in the net input is so sublinear that even after you then put the net input through the superlinear input-output function, the responses grow sublinearly, either linearly or sublinearly in different parameter regimes. But in a broad parameter regime, the responses now will grow sublinearly with the feed-forward input. And so you have this very generic transition from, so you have this very generic transition from a weak activation, largely uncoupled regime where responses sum superlinearly to a strongly activated, recurrently dominated regime where your responses add sublinearly. And that simple, very generic idea that all comes back to that you have some instability, namely the superlinear input-output function of individual neurons that needs to be stabilized by the network, that is going to now explain a wealth of data, as I hope I'm going to convince you. So let me show you a little bit more about how this SSN works. You get a little more intuition for it. So we have firing rate equations, which are just going to be very simple. We have a population vector R, which each element of this vector is the firing rate of one of these neurons. We have a connectivity matrix W, the matrix of all the connections between the neurons. And what we're saying is that the firing rate is moving with first-order dynamics towards what would be the instantaneous steady state, which is the recurrent input. W times R is all the recurrent input that the given neuron is receiving, plus the feed-forward input, which I'm going to call G for a shape. That is, this guy receives twice as much as that guy and half as much as that guy. The overall shape, but normalized to a length 1. And then a scalar, just a number C, saying how strong is my input, C for contrast. And so take my feed-forward input, take my recurrent input, rectify them, raise them to the n, and that's what's driving my current growth of, or change in my firing rate, so that at the steady state, my firing rate is just equal to this net input raised to the n. So what happens with this model? Well, in the weakly activated regime, so now this is C small. I haven't told you what small is, but take my word for it. This is the weakly activated regime. What you see then is here's the feed-forward input is just, by definition, it's linear in C. C is the strength of the feed-forward input. So here's the feed-forward input in blue. And the total recurrent input, you see, is much smaller, or smaller than the feed-forward input. It's growing, it's coming up, but it's smaller than that. And the excitatory response is just this plus that raised to the power n. I think it's 2 in this case. And so here's the excitatory response growing superlinearly as a function of the feed-forward input. But now as you get into the higher input strength regime, basically somewhere around 10, the excitatory subnetwork is going unstable. And so this is the little region I showed you before. But what you see now is happening is that the recurrent input is, it was sort of growing positive, so it was excitation dominated, it was positive, it was growing along with the feed-forward input. It starts to tail off and become inhibition dominated, and furthermore, it starts to grow negatively with exactly the same slope, or almost exactly the same slope, as the positive growth of the feed-forward input, so that the net input, the feed-forward plus the recurrent, is leveling off. In fact, it looks like it's plateauing, but when you square it and change the units appropriately to get the firing rate of the excitatory cell, you see that it is in fact still growing, but it's growing slowly enough that even after going through this power law, superlinear nonlinearity, the excitatory rate is growing sublinearly. And so this is what's happening, is the recurrence is basically conspiring to largely cancel the feed forward, leaving a net input that's growing much more slowly than the feed forward input. And so summation becomes either linear or, in a broad parameter regime, sublinear. And so this dynamic stabilization leads to a dynamic balancing of excitation and inhibition. That is, the net input is growing slowly, so your excitation and inhibition is largely balanced. I should say, well, I'll tell you in a minute, it's a weak balancing, not a strong balancing. So to summarize, for very weak inputs, we have superlinear response growth, which when we think about what the surround does, the context, that's going to be a regime where you can have facilitative contextual modulation. For moderately strong inputs, sublinear response growth, regime where you have suppressive contextual modulation. This is all in this paper we published in 2013, but you can show that this transition from superlinear to sublinear occurs when this dimensionless parameter alpha, which is basically the average strength of the recurrent weights times the strength of the feed-forward input raised to the n-1, when this dimensionless parameter alpha is order 1. So that's what we mean by big and small. And some of you may be familiar with the balanced network model, very well known, of Van Vrieswijk and Sampolinski, which was a really important model for theory, because what it showed was that that you could have a network that had extremely strong input that would dynamically stabilize itself with inhibition, and so would dynamically find a region where the net input was subthreshold and it was firing on the fluctuations, which gave you the variability that you see in Cortex, even though the excitatory input alone would have taken you way over threshold. And so this fact that you would dynamically balance in a broad parameter regime was a big breakthrough. But the connection to what we're talking about is that the balanced network solution arises in the limit in which this parameter alpha is going to infinity. Very, very large input. And in that limit, you can only get linear behavior. You cannot get any, the nonlinear contributions are negligible. The linear contribution just completely dominates. And furthermore, this gives a very tight balance. That is, you have very large feed-forward input, much larger than the distance from rest to threshold, almost precisely canceled by very large recurrent input, leaving a net input which is less than the distance from rest to threshold, so much smaller than the feed-forward input alone. But this is actually contradicted by a number of experiments from David Furster's lab and more recently from Scanziani and from Li Zhang and Tao that show that when you block the cortex and isolate the thalamic input to layer 4 in V1, that the thalamic input is smaller than the net input. It's not, an order of magnitude bigger. So that's a problem for the balanced network, as well as the fact that it only allows linear behavior. What the SSN does essentially is generalize this idea that you get the network dynamically stabilizes itself and cancels the feed-forward input, but this already begins when alpha is order 1. And at that point, it's a loose balancing. It's where the net input, the feed-forward can be smaller than the net input or comparable to the net input. They both can be less than threshold, so it's consistent with experiments. And you get all this nonlinear behavior, which I'm now about to show you, explains an awful lot of biology. Okay, so the biology this can explain, let me begin with normalization and stimulus-induced suppression of correlated variability. So actually, I've already been referring to contrast. I'm sorry, I should have defined this at the beginning, but define it now to make sure everyone's on the same page. What's the contrast of a visual stimulus? It's just the light-dark difference relative to the mean. So this is a low-contrast stimulus. This is a high-contrast stimulus. Its significance for us is that the lateral geniculate nucleus, the thalamic nucleus that's relaying information from the eyes to V1, primary visual cortex, the LGN firing rates are monotonically increasing with contrast. So contrast basically stands for the strength of our feed-forward input. So what is normalization? It's essentially, well, I should say normalization is a word that's been used to refer to every single thing about responses that is not a linear function of contrast. And it's a long story I could talk with you about later, but that's many different things, involves many different mechanisms. But the most dominant thing people mean by normalization is that the firing rates themselves sum sublinearly. And so this is illustrated here from David Fitzpatrick's lab, where they bin neurons by their preferred orientation. So here we're just seeing neurons arranged by their preferred orientation. They show these stimuli, either the 45 degrees or the 135 degrees or both at the same time, moving, drifting. And this is the response across neurons to the 45-degree stimulus. Naturally, the neurons that prefer 45 respond best. This is the response across neurons to the 135-degree stimulus by itself. When you show both together, you get the response that's shown in red, which is very close to the blue, which is the average of the two responses rather than the sum of the two responses. So you're getting a very sublinear summation. It's also shown here from Matteo Carandini's lab, again, where he's combining vertical orientations at varying contrast, horizontal orientations at varying contrast. And so this is now for two equal strength stimuli, but now this is showing you a cross strength of stimuli. First, if you go down the diagonal, you see that when they're both present at a given contrast, you get much less than the response to one alone at that contrast. But second, you see that as they become unequal, you get a winner-take-all response, where the stronger one basically dominates the response, is all that you see, and the weaker one just doesn't, which by itself might give this bigger response, doesn't even contribute to the response. I actually should have waited to push this up. Your attention should have been up here, but here we go. Unfortunately for our story, much of this cross-orientation depression probably is subcortical. It's not occurring in V1. It's occurring in the effects on the LGN and what that does to the summed set of LGN inputs that a given V1 cell receives. But there is a cortical component, because a weak component of this cross-orientation suppression, so in other words, you show the preferred orientation, you get a response. You show the cross-orientation, and you suppress the response. So it's called cross-orientation suppression. there is a component of this cross-orientation suppression which is binocularly mediated. That is, you can show one orientation in one eye, one orientation in the other eye, you still get some suppression. And that can only happen in cortex because all the LGN cells are monocular. So there is some piece of it in cortex. And furthermore, this normalization operation, this sublinear response summation, is ubiquitous across cortical areas, reviewed by Carandini and Heeger, 2012. It seems likely to be performed by the cortical circuit, even when the inputs are adding linearly. I have to say, we have very little. I'll show you one example from the surround later, but we have very little actual direct evidence that a piece of cortex is receiving two inputs that add linearly and then is sublinearly summing them in that piece of cortex. What we know is that you show two stimuli in the world and that piece of cortex sums them sublinearly. So although we don't really have definitive proof of this, everybody believes, and I believe, and I'm going to ask you to believe, that normalization is being done by each little piece of cortical circuit. And so what we're going to do is, whoops, I thought there was one more thing there. So I thought I had another. Anyways, what we're going to do is we're going to assume that the inputs add linearly in order to focus on what does the cortical circuit do when the inputs are adding linearly. What does the model cortical circuit do? But this is showing you that, so that was showing you normalization, a typical example of normalization, although it might be mostly subcortical. But when the stimuli are weaker, they don't add sublinearly. They add linearly or maybe superlinearly. So this is from area MT in the monkey, a higher visual cortical area. And this is showing you the response compared to the linear prediction when you show two stimuli in the receptor field, now here are different positions in the receptor field of this MT cell. They tend to be sublinear at high contrast, but what they call mixed contrast or what they call low contrast tend to be linear. What they define as low contrast is that the contrast less than that which gives 50% of the maximum response for the cell. That's actually not very low contrast. Getting 50% of the response is already pretty strong. And what they mean by mixed contrast is one of the two contrasts is low. What we see here is that on average, when at least one or both of them are less than 50%, you're getting linear summation. What we don't know is when it's really low, is that these superlinear points? Are you getting actually something superlinear when it's really low? A hint of that is here they plotted the iso response contours versus the two contrasts. And you see that for higher contrast, you have this concave, I always get them mixed up, shape. But for lower contrast, it flips and goes the other way. It's a hint, it's not a proof by any means, but it's a hint that you may be going from sublinear to superlinear. And I'll show you that our model, which goes from sublinear to superlinear, does give you these same shapes, although it doesn't prove it. At least linear summation for weak inputs is also seen in multisensory integration. In MST and VIP, superlinear is seen in superior colliculus. How we're going to model normalization? So we're going to model this cross-orientation suppression idea. So we're just going to consider cells to be characterized by their preferred orientation. And since preferred orientation lives on a ring, and I forgot to tell you, for those of you who don't know V1, the most salient thing you see when you study a V1 cell is that they really care a lot about the orientation of a light-dark edge. So they're very tuned for that orientation. There's some orientation they prefer, and as the orientation gets off of that in their little receptive field, the piece of visual space they're looking at, they really care about, there's some orientation they prefer and orientations away from that they don't respond to very well. So we're going to characterize neurons by their preferred orientation. Since that lives on a ring, then we can think of our neurons living on a ring where they are in the ring represents their preferred orientation. At every point, we're going to have an excitatory unit and an inhibitory unit, which may or may not correspond to a neuron. You can think of it as sort of a local collection of neurons. We know that in cat V1, at least, in layers 2 through 4, the excitation that the cell receives and the inhibition that the cell receives have the same orientation tuning. And so to model that, we're going to assume that the spread of connections from the excitatory cells and from the inhibitory cells all have the same shape. They're all Gaussian with about a 30-degree width, and they differ only in their relative amplitudes, these four kinds of connections, E to E, E to I to E, and I to I. As I said, we're going to assume the inputs add linearly, so we can study how the cortex processes linearly adding inputs. What do we mean by an input? When I show a given orientation, I'm going to assume I have a Gaussian amplitude, the input as a function of orientation is going to have a Gaussian shape, again with around a 30-degree width centered on the orientation of the stimulus. So first of all, do we see this transition from, we're going to have this power law input-output function. Do we see this transition? So first, focus on the left. What I'm showing you is this is the percent of the net input that cells receive, which is external, which is feed forward, and this is the percent which is recurrent. And then the red or orange represents the input to excitatory units, and the blue or cyan represents the input to inhibitory units. And what you see is indeed, for very weak drive, very weak input strength, this is for showing one grading, single oriented grading. For very weak drive, you're feed-forward dominated, but very quickly you reach this transition. I said it was around 10, but it's around there where the excitation goes stable about where unstable about where these two things are equal, but the recurrent quickly comes up, becomes equal to the feed-forward, and then comes to dominate. Furthermore, if you look at among the recurrent input, how much of it is excitatory, how much of it is inhibitory. So look at the ratio of the excitatory recurrent input to the total recurrent input. Where E plus I is represented by its absolute value. We're just talking about the net strength of the recurrent input. What you see is that for weak input, it's mostly excitatory recurrence, and it becomes more and more inhibition dominated as you increase the input strength. And this is very reminiscent of results from Dan Feldman's lab in mouse somatosensory cortex, where they optogenetically excited the E cells, and they record the excitatory and inhibitory input received by other E cells that weren't optogenetically excited. And what they see is as a function of how strong their light stimulus was how strong their optogenic activation was, initially they were just getting excitatory input, and then they were recruiting more and more and more inhibition with increasing activation level. So let me show you how this model cortex normalizes. So let's imagine now, here's all the neurons on my ring, but they're laid out on a line by their preferred orientation from 0 to 180 degrees. Here I'm showing a 45-degree stimulus, and so the response across the population is just a bump of activity centered on 45 degrees. I show 135-degree stimulus, I get a bump at 135 degrees. When I show them both, I get two bumps, but they're smaller. And if you can read these numbers, they're about 68% of the amplitude of the individual responses alone. And you can see that most clearly here, where red is the actual response. It's just this now is being shown in red. Blue is the average of these two responses. Green is the sum of these two responses. And so you see the actual response is something closer to the average than the sum. It's a sublinear response summation. Now, furthermore, and this may be surprising, this is what the excitatory cells are doing. People have often thought that normalization comes because there's a bunch of inhibitory cells that don't normalize. They linearly or superlinearly add, so they fire like crazy, and they make the excitatory cells reduce their responses and sublinearly add. But that's not what's going on here. The inhibitory cells are also normalizing. Everybody is normalizing. So here's the same thing for the inhibitory cells, and again you see that the response to the two together in red is closer to the average of the individual responses than the sum of the two responses. So what's happening here is that normalization is not because you've got a lot of inhibition that's driving down your excitation. It's a network effect where the network has to find a stable point. It's trying to, it wants to explode. It's stabilizing against that. It's pushing the inhibition down, excitation down. But when the excitation comes down, that removes excitation of the inhibitory cells, and they're coming down too. And the stable point at which you settle turns out to be a place where everybody is sublinearly adding. And there's no inhibitory pool that's firing a lot to do the normalization. And this, by the way, one of the problems with the inhibitory pool models is that people have looked for this excess inhibitory conductance and have never seen it. And this is now at least an explanation of why you wouldn't expect to see a lot of inhibitory conductance, because the inhibition is also being normalized. But this summation is contrast dependent. So if we model the response to two stimuli as a weight times the sum of the two individual responses, and then say, what is that weight? Well, it's nice and sublinear, stays around 0.7 or so across a broad range of contrasts. But at those very low contrasts where the feed forward was dominating, and right about here is where the recurrence equals the feed forward. For these very low contrasts, you're getting superlinear summation, as expected, because you're feed forward dominated, and your individual cells have superlinear input-output functions. I showed you before these iso-response contours. This is what they look like in the model. And again, you're getting a transition from concave to convex. In the model, this represents superlinear to sublinear transition, but it doesn't have to mean that in MT. There's other interpretations. The winner-take-all effect I mentioned to you, that also results in the model. So now what I'm showing you is the orange is the response to one stimulus, the green is the response to the other stimulus alone, and the black is the response to the two together. This is what I just showed you when they're of equal strength. I'm getting this average, this sublinear summation. As their strengths become more and more unequal, you see that the weaker one is getting more suppressed than the stronger one until eventually you're just getting the strong response and you don't see the weak response at all. And so if we model that by taking the response to two stimuli as one weight times the response, this is across the population, so it's a least squares fitting. The response across the population to one stimulus weighted by W1 plus the other stimulus weighted by W2, then the weight on the stronger stimulus goes to 1, and the weight on the weaker stimulus goes to 0 as the strength of their activations gets more and more unequal. And you can actually show that if you're doing sublinear summation, then this winner-take-all effect is just a consequence of the fact that you're doing sublinear summation. So this normalization and winner-take-all can explain another effect, which is the reduction of shared or correlated variability with the onset of a stimulus or plan or attention, any sort of increase in drive to the neuron. This was not for attention, but for stimulus or a motor plan onset, was shown in a paper with 1 million authors with Mark Churchland organized, where across, here I'm showing several visual areas, V1, V4, MT, but also across motor areas. When they look at the Fano factor, which is the variance of the spike count in some time bin over the mean in that spike count. So it's a measure of how variable are the spike counts from trial to trial. When they look at the Fano factor, at the onset of a stimulus, the Fano factor goes down consistently across these areas. And they showed that this wasn't just an effect of the fact that the stimulus was driving the mean rates up, because when they looked in the network at neurons that weren't going up, like if I'm showing horizontal neurons that are selected for vertical, their Fano factors went down too. So it was the whole network was lowering its variability, and not just the ones that were being driven as you drive the network. And that results in the model. I guess before we go through the slide, I'll just tell you intuitively why does it result. There's sort of two intuitions. One is the winner-take-all effect. If you think of the stimulus as one input, or the mean drive as one input, and the fluctuations as another input, and they're competing. As you make the mean drive stronger compared to the fluctuations, this winner-take-all effect means that the fluctuations contribute less and less to the neuron's output. The other way to think of it is that you start out for very weak input with the whole network stable, the excitation, you could take away the inhibition, the network would be stable. And then as you drive the network up and the excitation first goes unstable, it turns out that at that point, the network as a whole, the network stays stable because of feedback inhibition, but at that point, the network as a whole comes as close as it's ever going to get to being unstable. And then as you drive it further into the inhibition stabilized regime, it kind of backs off further and further away from being unstable. Mathematically, it means if you linearize around the fixed point, your eigenvalues are as close to 0 at that point as they're going to be, and then they start getting more and more negative again. Well, as the eigenvalues get more negative, it means you're doing less amplification of your fluctuations, and that's driving down the noise. So I'll show you this now. This is the same ring model. This is just showing the connectivity, which I explained to you before. And what you're seeing here is the firing rates, firing rates from 0 to 80 hertz on the color bar across the network. Before a stimulus, and now we're giving some random noise to the network so that we have some stochasticity to talk about variability as well as the stimulus. So here we don't have the stimulus, just the random noise. And then we add the stimulus, and you see this focusing of activity. But the activity, if we look at the neurons who prefer the stimulus that we're showing, here's their firing rates going up, and here's their Fano factor going down. And the neurons that don't prefer the stimulus, again, here's their firing rates not really changing, and here's their Fano factor going down. And furthermore, Churchland et al. Showed that if you do a factor analysis to separate the neuronal variability into the shared component, what's correlated between neurons, and the private component, each neuron's individual Poisson, presumably, variability, that what's being suppressed when you add the stimulus is the shared component, and the private component really isn't being touched. And that's also true in the model. And again, there's sort of two ways to think about this. One is that it's not a single cell effect. This is a network conspiracy that is suppressing the variability. And so the network has got to see the variability. And to the extent to which it's private to individual neurons, it doesn't add up to much on other neurons, and the network basically doesn't see it and doesn't suppress it. The other way to think of it is that now you've got a spatial network. You've actually got different modes. You can think of, say, the Fourier modes if you want. You've got different modes, so some broad excitation and some little fine going up and down. And some of those modes are unstable, and some of them are being dynamically stabilized, and some of them are not. And it's the broader modes that are being dynamically stabilized, and they represent the shared variability. And so this also explains the fact that you're suppressing shared variability means you're suppressing correlations. And as some of you may know, it's been a big topic recently that when you attend to something, it suppresses the correlations as well as the variability in your activity. And this, if you think of attention as adding an input, then this explains why that happens. Okay, so that's normalization, stimulus-induced suppression of correlated variability. What about surround suppression? So let me just tell you what surround suppression is. We've alluded to it a lot, but so here's an example from V1. We're showing a grading that's drifting along. Here's a static snapshot, but it's drifting. Within this window, there's the white and black bars are moving within this window. Blank screen, here's the firing rate. So what this x-axis is across one cycle of the drifting stimulus. And so here across time, across one cycle, we're just getting some random activity when it's blank. I show the neuron's preferred stimulus of its preferred size, and I'm getting a big response when at half the cycle, basically when the light's in the right place for the neuron and the dark's in the right place for the neuron. Now if I make the stimulus bigger, I get a much smaller response. And furthermore, this suppression depends on the orientation of the surround relative to the center. When the orientation is when the surround is at the opposite orientation, I don't really get very much suppression. So again, the center being the region where appropriate light stimuli can drive spikes. The surround stimulus, you can't drive spikes from there by itself, but they suppress responses to the center stimulus. This is tuned for stimulus orientation. And this surround suppression has been seen basically at least in every visual area it's been looked at. You see something like that in other auditory and somatosensory areas where it's been looked at, but it remains unclear how much of that's already in the thalamus and how much is in cortex. But in V1, at least we know this is really being done in cortex. And wherever it's been looked at least these three areas, when the stimulus is of low contrast, when the center stimulus is of low contrast, then a high contrast surround either can facilitate your response rather than suppress it, or more weakly suppress your response than it would when the center is at high contrast. The idea that the surround enhances at low contrast and suppresses at high contrast, you can look at the length tuning curve. So as I make the stimulus bigger and bigger, what's the neuron's response? So the neuron response goes up to some optimal length, and then the neuron is getting surround suppressed, and so its response is going down. And this peak, the optimal size, we call the summation field size. And what's been shown, this is in monkeys, is that if you look at the ratio of the low contrast summation field size to the high contrast summation field size, it tends to be much bigger than 1. Here it's about 2.5 or something, but it all depends on what you call low contrast, and they didn't really go to that low contrast. But so we have a sense that summation field size is monotonically shrinking with contrast, although not that many points in that have really been filled in. But and so what that means then is that when your center is high contrast, high contrast surround suppresses you, but when your center is low contrast, stimuli in that same region is facilitating you, low contrast. Furthermore, when you consider now that the center and surround are different contrasts, a high contrast surround can enhance response to a low contrast center, but suppress response to a high contrast center. And this is data from Seung in Cat V1. This is now the center contrast on this axis. This is the neuron's response. This is the response to the center stimulus alone versus its contrast. Now you add a 10% contrast surround, or you add a 40% contrast surround. And what you see is when the center contrast is less than about 15%, the surround is enhancing the response. And when the center contrast is stronger than that, then the surround is suppressing the response. This is not universally seen, and we have an explanation for that, as I'll tell you in a minute. But at least in some circumstances, you actually get this change to facilitation. Other people just see the suppression get weaker. And Matteo Chiarandini, more recently in the mouse, optogenetically activated input from the surround to the neurons that he was recording from. And he saw this same effect, that as he increased the power of the light stimulus that was activating the surround neurons, so this is the response to the center alone as a function of the center contrast. As he increasingly strongly activated the surrounding neurons, the response went up at low contrast, but at low center contrast, went down at high center contrast. So to tell you about our models of surround suppression, I want to break this up into a couple of pieces. First, I want to review work we published in 2009 on what we call the inhibition stabilized network model. Here we just showed that the data on surround suppression showed that the network had to be in this inhibition stabilized regime where the excitation alone was unstable. But that could happen in a linear model. We weren't yet dealing with any nonlinearities. So we'll start there. So what's the likely mechanism of surround suppression in V1? Well, the center is activating some region of cortex, maybe about a millimeter in size. And the surround is activating adjacent regions of cortex, millimeter or several millimeters away. The only communication that goes across millimeters in cortex are the so-called long-range excitatory projections. So only excitatory projections traverse those kind of distances. And also you can go through feedback to higher areas, go up to V2 and back to V1. That will also, but again, that's purely excitatory input. So the only way to get across long distances in cortex is with excitatory projections. It's always been imagined that to cause suppression, this input has to preferentially drive the inhibitory cells. And the picture everybody has had is that why do cells get surround suppressed? Because you're getting this excitatory drive to your inhibitory cells, so your inhibitory cells are being surround facilitated. They're firing more, and that's causing your excitatory cells to get inhibited, and so they're firing less. And so the expectation, So the expectation is that if you looked at the inhibition the cell was receiving, when you added a surround, it should be receiving more inhibition, and that's what's making it be surround suppressed. Well, David Furster did this experiment, and this is actually the experiment that started us on the long trajectory to what I'm telling you about today. Did this experiment, and he surprisingly found that both the inhibition and the excitation that cells receive decrease when they're surround suppressed. So he compared the steady state response to an optimal size stimulus to the response to a very large stimulus. It's typically about 2 degrees. It's typically about 20 degrees diameter. And this is the excitation that they receive. And you see that when you add the surround, they receive less excitation than when you add the center alone. When you add this is the inhibition they receive. And when you add the surround, they receive less inhibition also than what from the center alone. And so basically the proximate cause of being suppressed is not that you're getting more inhibition, it's that you're getting less excitation. And that the decrease in your excitation exceeds your decrease in your inhibition enough that it's suppressing your response. So the interpretation that we made is that both excitatory and inhibitory cells are being suppressed. This was later confirmed in CAT V1 by a Chinese group who identified excitatory and inhibitory cells, showed both being surround suppressed. And in mouse V1, it's kind of been controversial. There's a very prominent paper from Scanziani lab saying that one class of inhibitory cells was surround facilitated and claiming that that increased inhibition from those cells was probably driving the suppression. I'll get back to that later. But 2 other more recent studies say, no, the inhibitory neurons in mouse V1 are also surround suppressed. And there's a whole lot of evidence. You might say, well, maybe one explanation for this would be, I'm just suppressing the LGN input. If I'm giving less feed-forward drive, then everybody, my excitatory and my inhibitory cells, all get less drive. They all fire less, and everybody receives less excitation, less inhibition. So one way to explain this would be that the local circuit, the surround is causing the local circuit to get less excitation. We can rule out that this is due to an effect on the LGN through a variety of things. One is the orientation tuning of the surround suppression, which you wouldn't see from an LGN effect. But there's a lot more that shows that the main component, there is a small untuned component of surround suppression that is due to LGN, but the main orientation tuning component is coming from cortex. So what we showed in this 2009 paper is that assuming the surround stimulus doesn't increase excitatory input to the local circuit, like LGN input, then this can only happen if the network is inhibition stabilized, which we call an ISN, inhibition stabilized network, meaning that the excitatory recurrence would be strong enough to be unstable, but the network is stabilized by feedback inhibition. And this leads to a paradoxical result, which I'm going to tell you about, that actually Misha Sodiks had first identified in 1997 in a model of hippocampus, which we didn't know about and had to reinvent, and then realized we had rediscovered the beautiful thing Misha had found. And so let me just show you how this works. And there's nothing about nonlinearities here. This is just about being inhibition stabilized, however you get there. Before I showed you that with this nonlinearity, you will get there, but however you get there. So imagine we're just going to have a very simple model, a population of excitatory cells, a population of inhibitory cells. Excitatory cells excite themselves and the other guys, and the inhibitory cells inhibit themselves and the other guys. And we imagine that we're getting both a center stimulus and then we're going to get a surround stimulus. So first of all, just imagine we're only getting the center stimulus. We have some overall, we're getting some fixed level of center input. We have some fixed point of response, represented by gray, to that center input. And now we're going to add the surround stimulus. So here I'm just going to show you the surround being added, but the center is always there. We're perturbing that fixed point, that center fixed point. So what happens when I add the surround? Well, I'm adding drive, and to simplify, I'm going to say, let's suppose the surround only goes to the I cells. Really, it goes to both, but we have to imagine it's dominantly affecting the I cells to cause suppression. So imagine it only goes to the I cells. So then it increases the firing rate of these inhibitory cells, and that lowers the firing rate of these excitatory cells. And that's going to withdraw. So I've added excitation to the inhibitory cells, but I'm also going to withdraw excitation from the inhibitory cells. But now, what happens when I lower this firing rate? Well, if the network if the excitatory subnetwork was unstable by itself, what do I mean by that? I mean that I'm at the center-only fixed point, and now I freeze the inhibitory firing. Not that I eliminate all inhibition, but I freeze it at the fixed point level. I don't let it dynamically respond anymore. And I say, in that case, if I perturb the excitatory firing a little bit up, would it keep going up? Or if I perturbed it a little bit down, would it keep going down? In that case, the excitatory subnetwork by itself is unstable, but then when I let the inhibitory cells dynamically respond, the whole network will be stable, and I'll be restored to the fixed point. So if I'm unstable by itself, if the excitation is unstable by itself, what it means is that when I if I were to tick the excitatory firing rates up a little bit, freezing the inhibition, I would recruit too much recurrent excitation, and they would therefore want to keep going up. But it also means if I tick them down a little bit, I lose too much recurrent excitation for that lower firing rate, and I want to keep going down. So now what's happened? I've lowered the excitatory firing rate, so I've lost too much recurrent excitation to even support that lower firing rate. And not only that, but I've got more inhibition than I had at the fixed point, which also means I want. So I'm in free fall. My firing rate has started to fall, and it's going to keep going down. There's nothing to stop me until this withdrawal of excitation gets bigger than the excitation I added to start the process. At that point, the inhibitory cells are receiving less excitation than when they started. And so now their firing rate becomes lower than when they started. And so you might say, OK, now the excitatory cell is receiving less inhibition than when it started, so it's going to go up, and the whole thing's going to oscillate. But no, remember that this guy firing less has too little recurrent excitation. The only way to stabilize that lower firing rate is for it to receive less inhibition than it was in the previous fixed point. And so this lowering of the inhibitory firing rate actually stabilizes the new fixed point. And this is the new fixed point of the system. Everybody has lowered their firing rate, both E and I. It's not the case, just as for normalization, it's not the case that the inhibitory cells are firing a lot to the surround, which is making the excitatory cells go down. Everybody is self-consistently being surround suppressed. That's how the network finds its new stable fixed point. And Misha Sodiks called this the paradoxical effect, that when you try to add excitation to the inhibitory cells, they lower their firing rate in the new steady state. The key prediction of this is that, so David showed in the center-only fixed point, I get a certain amount of inhibition, and in the surround plus center fixed point, I'm getting less inhibition. What he didn't do was look dynamically. Is there a moment in time when I'm getting more inhibition before I start getting less inhibition, which is what this model predicts. So he went back, Hirofumi Ozeki went back, and spent a year getting these time-resolved conductance measurements. But indeed, they saw the transient increase before the steady-state decrease. And so I'll just show you that. Here's now, having only the center stimulus, I'm showing you here the voltage at And the way they reconstruct excitatory inhibitory conductances is they do the same experiment at 0 holding current and minus 250, minus 500. And so you're getting different voltages. And by seeing how the voltage changes at each point in time as a function of the current, you can at least if it were a point neuron, it's a little dicey since it's not a point neuron, but at least assuming it's a point neuron, you can reconstruct what the excitatory and the inhibitory conductances were as a function of time. And so what you see here, now we add this around, and you see for this neuron, both the excitation and the inhibition go up before they go down in the new steady state. So the excitation is in red, and the inhibition is in blue. When I show the orthogonal surround, I don't get that transient increase. Nothing goes down. Both conductances go down a little bit, as the firing rate doesn't change much. Here's another neuron. Here, only the excitation goes up, not the inhibition. But again, I get the transient increase before the steady state decrease. Doesn't happen for the orthogonal. Now, these are complex cells, meaning that they don't care about the phase of the grading. As the grading drifts along, they're just responding steadily. This is a simple cell that only fires when the light's in the right place. So it only fires on one half of the cycle as the grading drifts across. And so you see its voltage going up and down as the cycle drifts, as the grading drifts. And so you can now add this around at various stages in this cycle, when the response is going up, when it peaks, when it's going down, or when it's at the bottom. And no matter which phase you add it at, you always see this transient increase in the inhibition received before the steady state decrease. So to me, this is the smoking gun of this inhibition stabilized scenario. You could maybe cook up some other scenario where you have a biphasic change in inhibition, but it's hard for me to think of one. Okay, so that was our ISN model. And actually, the very first thing we did was just extend that to that was just one excitatory, one inhibitory population. We wanted to extend that to a spatial model where the center and surround, I won't be too much longer, where the center and surround are both self-consistent parts of the same circuit. But we begin with just a linear model, which is going to reveal one important piece of behavior. And so the model is, I have now neurons, excitatory and inhibitory neurons arrayed on a line, representing retinotopic position. I assume these are all neurons preferring the same orientation, because those are the ones that connect to each other by the long distance connections. That's why the surround suppression orientation is selective. And for a drifting grading stimulus, I'm going to imagine a uniform input to all the neurons retinotopically seeing that stimulus. Uniform because either assuming that these are complex cells in layer 2-3 that don't care much about the grading of the phase of the grading, or they just get a uniform input, or you can imagine we're looking at an ensemble of excitatory cells that prefer different phases that are exciting one another. And the connectivity is, we assume, as is known in Cortex, that the excitatory projections project long range, the inhibitory projections are very local, actually only projecting within one point here, but we don't need that, but that they're local, there's a long range. And we do a funny thing, which is we assume that the E to I is longer range than the E to E. It doesn't have to be physically longer range than this, it just means that the longer range connection should be more biased towards I than the shorter range connections, and that would be enough. We don't know if that's true, but if it is true, we get an interesting thing, which I'm going to tell you about. And so our model is now, it's a linear model, so that the input is just a linear function. It's just the sum of all the different input contributions, the feed forward and the recurrent. And what happens now is that we get periodicity in spatial activity and in tuning. So conceptually, I'm showing a bigger and bigger grading. What I'm actually doing, it's a one-dimensional model. I'm actually showing this pattern of input across the one-dimensional line of cells. So this is the actual input. This is the response across the excitatory population on the line, and this is the response across the inhibitory population on the line. And what you see is that the response, and this is for high contrast grading, well, but for a linear model, it doesn't matter. Response is the same at any contrast. What you see is that you're developing this periodicity, and the periodicity is roughly the same for E and I. They're not actually exactly the same. But what's happening is the network is developing these resonant frequencies where it most amplifies the input, basically. And so that's what's coming out in the activity pattern. And this relies on the E to I being wider, or the long-range projections being a little bit more tilted to I than the short-range projections, to give you some sort of Mexican hat structure to get you started with this. But what this means is if we now look at length tuning curves, if you consider the cell, there's one other thing I want to focus your attention on, which is you notice the peaks of the activation are always at the edges this was pointed out in an old model by Misha Sodiks also. The peaks are always at the edges because the edges are not getting surround suppression from one side. So they always have the peaks, and that means the phase at the middle is going down and up and down again. Because you've got a fixed period and it's getting bigger and bigger, and the peaks are always at the edges. So if you now look at the length tuning of the cell in the middle, it goes up, and then it starts getting surround suppressed, but now the phase goes up again. And so if you look at its length tuning, it gets a second bump in the length tuning curve, both in the excitatory and the inhibitory cells. I should tell you, truth in advertising, this starts at 0.4. This does not start at 0. This is pretty weak modulation in the inhibitory cells, at least for these particular parameters. But it's there. And one thing that this can at least qualitatively explain, doesn't quantitatively explain, is in an old paper of David Furster's, where they looked at length tuning rather than sort of a disk increasing in size, just a preferred bar increasing in size. What they saw was that the inhibitory conductance went down when the cell was length suppressed, which is the same effect that later got us attention. That went down when you're surround suppressed, but none of us noticed this. But the main thing is that they got a second peak at further length, which they assumed, oh, when I make it big, I'm activating some new source of inhibition. And we're saying, no, you're just activating your center is just going up again as you make your stimulus longer. And at least qualitatively, that would account for that. And we can also show that you have to be in the inhibition stabilized regime to get the inhibitory activity to be spatially periodic in the absence of periodic connectivity. Okay, so now what does the SSN model add to this? So first of all, we get the same behavior as before. As we increase the input strength, the feed-forward contribution, relative contribution goes down, the recurrent relative contribution goes up, and the recurrent contribution is more and more inhibition-dominated. But what happens is now, for very weak stimuli, there's no oscillation, there's no resonant frequency, or the resonant frequency is a 0 frequency, and there's no surround suppression. And then as we increase the strength of a stimulus, we're getting the beginnings of spatial period. So what I'm showing you, this is the activity across the network to a large stimulus of the given contrast, and this is the length tuning of the cell in the middle. Response as a function of stimulus length. And this is for the excitatory cells, and this is for the inhibitory cells. So we're seeing the spatial pattern of activity across the neurons and the response of the one neuron in the middle across length. And what you see then is that periodicity is developing in the spatial pattern, and you're starting to get surround suppression and starting to get second peaks. It's happening in both the inhibitory and the excitatory cells. You might notice that the inhibitory summation field size is bigger than the excitatory, which is something that's seen in Cortex. But notice the summation field size, the wavelength is getting higher with increase. So the wavelength now becomes dynamical. Even though you sort of built in some wavelength with this Mexican hat kind of structure, that isn't really fixing it. It's dynamically being shaped by the overall activity level, and it's shrinking with increasing the wavelength is shrinking with increasing contrast. And as a result, the first peak, the summation field size, is shrinking with contrast, as we know actually occurs. And so this now explains, I showed you before this second peak in inhibitory conductance. They saw that at high contrast, but they didn't see it at low contrast. Again, at least a qualitative explanation for why this would be a contrast-dependent second peak. I showed you before that the summation field size shrinks with contrast, and that's what happens in the model. So if you wanted to shrink by a factor of 2 or 3, you basically have to define low contrast to be somewhere around here, which is pretty consistent with what they did. This is how much it shrinks, where we set the asymptotic size to 1. I showed you about this experiment, the surround facilitating for weak and suppressing for strong. That's just what the model will do for a surround that's not too big, high contrast surround. Then here's the center-only response, and as we add the high contrast surround, it facilitates for the weak center, suppresses for the strong center. However, when the surround is large, then low contrast just gives you weaker suppression, doesn't go all the way to facilitation. And that might explain maybe why different labs get different results on this question. It might have to do with the shape of the stimuli they're using. And finally, do I have time to go to, or should I wrap up without the experimental tests? Sure. Okay. So Dan went to Steve Van Hooser's lab to test this model. And the first thing he tested was is there periodicity in the size tuning? He was working in anesthetized ferret V1. And so people usually haven't, I mean, you've seen hints of this periodic size tuning in the literature, but in fact, one paper that somewhat systematically looked at it, but people generally don't look at very fine resolution. They look at sort of around the summation field size, around the peak, and then they look far out where you're suppressed. So he looked at every degree from 1 degree to 30 degrees, randomly interleaved stimuli, and looked at the length tuning curves. And this is 6 example cells. And what you see is there are these second peaks. Here's the fit of 2 different models. One is just a standard difference of Gaussians, one Gaussian for the center, one for the surround in red. And the other is we add a couple of parameters that let the surround have a single frequency of oscillation, which we fit. And you see that, and that's in blue, and you see the fit with the frequency of oscillation in the surround is much better than without it. But by using a nested F test to compare the fit of these 2 models, which is supposed to take into account the extra parameters, nonetheless, the model with the extra parameters gives a significantly better fit in 72 of 76 cells, showing significant periodicity by this test. We also did cross-validation tests that showed similar results. So indeed, there is this periodicity in the length tuning, at least in a ferret. We'd like to see periodicity in the positions. We'd like to see spatial periodicity in the activity. So, you know, to a drifting grading in one place, these neurons are more active, these are less active, these are more active. That's a hard experiment to do because we only record from one neuron. So what you'd like to do is, you know, record from all these different neurons. But instead, we record from one neuron and move the grading around to randomly interleave positions. And the idea is that, you know, for some positions, the neuron is at a peak, and for other positions, it's at a trough, and it should vary periodically with the position of the grading. And that's exactly what you see. Here's 4 sample cells for position tuning, same everything as before. And 66 of 76 cells showed significant periodicity for the position of this drifting grading. And if you think of a receptive field, as most of us do, as something that just monotonically falls off, it's kind of surprising that as you move the position of a grading, you go down and you go up again. But that's what we see, and that's what this resonant frequency would predict. But now the key test of the SSN model is something that has to change with contrast. These wavelengths grow shorter with contrast. And so to study the shrinkage with contrast of the spatial period, problem is the tuning curves at low contrast are very noisy. And so we use a different assay of the periodicity of the network activity, which is called contrast modulation tuning. And this was studied by Izumi Ozawa and Tanaka in 2009. The idea is you show a very large grading covering center and surround of the neuron that's optimal for the neuron, optimal orientation, optimal spatial frequency, and that's drifting along. That's the black and white bars that you see drifting. And then you superimpose over that a slowly drifting contrast modulation, sinusoidal contrast modulation, at some orientation of frequency. So what that means is that these regions of cortex are being stimulated and these regions of cortex are not being stimulated. So you're periodically varying which regions of cortex are being stimulated, and you want to say what's the optimal spatial frequency of stimulation across cortex, and also what's the optimal orientation of this contrast modulation grading. And what they found was that, so the different colors represent different orientations of the contrast modulation. Remember, the luminance grading is always fixed at what's optimal for the cell being studied. And so you see that the response is tuned for the orientation of the contrast modulation, and it's also tuned for the spatial frequency. So it looks, basically the center plus surround of the receptor field looks like a big simple cell. It wants to see a certain spatial frequency where you are and you aren't stimulating, and a certain orientation of that. And the spatial frequency was typically sort of 1 to 6 times in their hands, 1 to 8 times in our hands, lower frequency or longer wavelength than the preferred luminance grading frequency of the cell. So how do we explain this? Well, our interpretation is that the preferred contrast modulation spatial frequency is the network resonant frequency. You get the best response out of the cell when you're hitting the cell at its peak and you're hitting the surrounding piece of cortex at the peak of the resonant frequency. Then you're really driving that resonance up, and you're not hitting the places in between, which are troughs, which would be suppressing you. The preferred contrast modulation spatial frequency is about 1 to 8 times lower than the preferred luminance spatial frequency. We can give the following explanation of that. The summation field size, I'll draw my one picture. So here's your resonant frequency, the optimal activation across cortex. The size of a stimulus that best activates a cell should activate about this much of cortex, because if it goes further, it's starting to get into the suppressing part of the resonance. And so this says that the summation field size, which is the diameter here of an optimal stimulus, shouldn't be any bigger than about half of my network frequency, my network wavelength. So the summation field size should be on the order of, or less than or on the order of half of a contrast modulation cycle, preferred contrast modulation cycle. And empirically, we know that the summation field size ranges from 1/2 to 4 luminance cycles. Put those together, you get the factor of 1 to 8. Why is the preferred orientation of the contrast modulation independent of the preferred luminance orientation? Well, basically, the preferred luminance orientation is saying what stimuli out there in the world drive the most input to me? What's the tuning of my feed-forward inputs? But the contrast modulation preferred orientation is saying, when I get this resonant activity across cortex, what direction across cortex is it most strong in? Well, that has to do with sort of the random irregularities in what you might imagine would otherwise be circularly symmetric connectivity. You break the symmetry, you pick some direction across cortex where you amplify your resonance most strongly. That's got nothing to do with what luminance orientation in the world is giving you the most input. And so these two things are uncorrelated with each other. Right. So show you how the model behaves. In the linear model, the contrast modulation stimulation, the spatial frequency that's preferred for contrast modulation is precisely where the spatial activity peaks, and precisely where we analytically solve is the resonant frequency. Don't just take my word for it. Don't try to go through all the details of that. And in the nonlinear model, the network resonant frequency grows with contrast. The preferred contrast modulation spatial frequency grows with contrast, more or less exactly as predicted by the growth of the resonant frequency with contrast. Is that no, you just put an S switcher. Okay. So in the experiment, here's 3 example cells. And in blue is the lowest contrast luminance. So we're varying the contrast of the luminance grading and then adding this contrast modulation, which goes from maximal contrast to 0. And so here's the lowest contrast of the luminance grading. And you see that, at least for these 3 cells, it's a low-pass tuning. They don't want to see any contrast modulation. They want 0 frequency. They want all of cortex stimulated at once. And this was true for about half the cells, that at the lowest contrast, they were low-pass. They preferred the lowest frequency. But as you go up in contrast, you're preferring higher and higher frequencies, not always monotonically higher for every cell. But if we average across cells, as we increase the stimulus contrast, the preferred contrast modulation spatial frequency is going up. Or another way to look at it is, if we just compare the highest to the lowest contrast, what spatial frequency do they prefer? 72% of cells prefer the highest frequency at the higher frequency at the higher contrast versus lower, and only 16% prefer the opposite. So we're seeing that by this measure, the network resonant frequency frequency is growing, wavelength is shrinking with contrast, as predicted. And if we look at these 3 different measures of the network resonance frequency, the preferred spatial frequency of the size tuning curve, the preferred spatial frequency of the position tuning curve, and the preferred spatial frequency of the contrast modulation for a high contrast stimulus, compare that to the luminance preferred spatial frequency, all 3 measures fall in the range sort of 1 to 8 times as the hand waving argument predicted. And interestingly, in the experiment, you'd think that these 3 measures would be correlated with one another. If one neuron had a longer wavelength size tuning, should have a longer wavelength position tuning and a longer wavelength contrast modulation preferred. It's not true. They're uncorrelated with the other experiments. It turns out when we go to the model and make the connectivity random, they're uncorrelated in the model as well. Basically, each paradigm is giving you kind of a different subnetwork that's being activated, and they're just uncorrelated with each other in what direction across cortex in what exact frequency they prefer across cortex. We can build a big 2-dimensional model with stochastic connectivity, and you get all the same behavior with lots of biological-like variability. I won't go into detail on that. I'll skip this in the interest of time, but there's a cool test. Look at figure 8 of our 2015 paper for this cool test. Any of you who work with channel adoption might be interested in this. This model can also explain multiple aspects of attentional modulation. There were some kind of cosmic confluence of, in 2009, where all of these groups published papers showing that if attention increases the input to the cells representing the attended stimulus, but into a circuit that does normalization, that would explain this huge array of effects of attention on neuronal responses. So naturally, you'd expect the SSM will do that, and it does. And also, they didn't address the fact that attention suppresses correlations and variability, which the SSN also accounts for. In the interest of time, I'll leave this out. I'll just say there's very unclear and both positive and negative evidence as to whether this applies to rodents, and I think it's an open question. I'd be glad to talk with you more about that. So in conclusion, suggesting that this basic motif, that just basic idea that given something, the superlinear nonlinearity that's driving you to instability, and that you then get dynamically stabilized against it, you get this transition from superlinear to sublinear behavior. You put that in the context of simple models of connectivity and explains a wide variety of cortical behavior. So is this a canonical cortical computation that describes the within-layer multi-input integration? And what is it telling us? Well, how should a layer integrate its various bottom-up and top-down inputs of varying strength across cortical space and time? Well, the SSN gives you different answers. It can produce winner-take-all, which is like a max pooling. Only the strong input contributes to the responses. But in other circumstances, the strong input can facilitate a weak input, as in surround facilitation. In other circumstances, you get more or less, for more equal strengths, you just get sublinear summation. But you can also get superlinear summation when the inputs are weaker. So different circumstances, you do different things in this, whatever this overall canonical computation is. And so it's suggesting that there's a more general flexible integration operation behind the observations with laboratory stimuli of normalization and surround suppression. And so obviously high on our agenda now is to try to address, well, if we've really captured something about cortex, which is an if, but if we have, what's the function of this flexible integration? And in particular, if you show natural stimuli to this circuit, to this motif, what does it do? How does it integrate the varying inputs across space and time in a natural scene? And one way to get at this, many of you are familiar with the recent excitement in deep networks. Deep networks use a max pooling operation. They use a normalization operation. Could we substitute this in there? And if we got really lucky, would it make the performance better? And if it did, and we got really lucky, then we would try to investigate, well, what is it changing about the processing, and what might be the computational function? It's a long-range goal, but that's what we'd like to understand. Thanks. And the people who did the work.
So we'll take questions, but I'll say that why don't we plan on sort of starting the next talk at 5 till, which is about 15 minutes from now. So those of you who need to have a quick stretch of the legs can do so. But in the meantime, those of you who have some questions, we can take some of those too. So somebody, good.
I think you whizzed past this on one of your last slides, but I'm wondering whether you are able to find these sorts of networks in primates, not mice.
Well, you see all these same phenomena, the surround suppression, the normalization, the suppression of variability as measured by the Fano factor with the addition of a stimulus in primates. It's been much harder to do circuit-level studies in primate. People are barely getting the opsins, the optogenetics to work in them, and up until very recently, people haven't been able to get intracellular recording to work in them, which is one way to tease apart what the excitation and inhibition are separately doing. So we don't know much about what's going on at a circuit level there. There was a recent paper from Nick Priebe, who for the first time got intracellular recording going in a awake-behaving monkey. And he saw a suppression of low-frequency variability when you added a stimulus compared to, so in the I'm trying to remember the details, but he saw what he thought was sort of a change in regime to a more balanced regime as he added a stimulus. And I'm not sure it was, but the one thing that was clear was there was some kind of suppression of variability at the voltage level, I think, going on in his intracellular recording. So we haven't specifically modeled that experiment yet, but I'm pretty sure we could account for that experiment with this model. Yeah.
So correct me if I get any of the details wrong, which I probably will. But I think you said that as you move from strong input, the network will move towards instability, but then backs off.
Yes.
It gets there.
Mm-hmm.
Presumably because of sublinear activation.
Well, basically, I mean, you're being stabilized by the inhibition. You know, at the point where the excitation wants to take off, it's driving up the inhibition, and the whole thing is finding a new settling point. And basically, the harder you drive it, you know, I don't have a good rigorous explanation for this, but basically, the harder you drive it, the more the inhibition stabilization is backing you off from the excitation going unstable. So
my question is, let's assume for some reason that the network does move into instability, past point of no return. Could that describe a ground mount seizure?
Yeah, absolutely. I mean, the fact that you get seizures with reduced inhibition certainly suggests the excitatory subnetwork is unstable by itself. There's a subtle point, though, which is that you remember when I defined in the surround suppression case what I meant by unstable, excitation being unstable by itself. I didn't mean in the absence of inhibition. I meant I'm considering some fixed point to some input, and then I freeze the inhibition at that level. And is the excitation still unstable there with that frozen inhibition? So it's not quite the same issue, but at any rate, you would certainly, the excitatory subnetwork going unstable seems to be what's going on in seizures. Yeah.
Is it possible, because it means the seizure could be reduced by, like stroke, for instance, which is a really intense contrast stimulus. Could it be possible that it's just so intense and it occurs so quickly that there isn't enough time to switch from supralinear to sublinear?
Or, yeah, absolutely. Or it just might be that, you know, you're operating near the edge of instability and you manage to drive some strong, fast fluctuation that just the inhibition can't take care of and can't, you know, and then so there is some route to instability. And I mean, for most people, strobe doesn't drive us there. Most of us have, you know, more stable brains, but for people who are prone to epilepsy, they can be right on the edge, and then, you know, although they stay stable under most circumstances, there can be circumstances that drive them off into instability. In this model, we would say that in those circumstances, that's a fluctuation that the inhibition just isn't sufficient to stabilize against. Yeah.
Do you require an equal number of excitatory and inhibitory?
No. So in all the models I showed you, we have equal numbers. That's just for computational convenience. But basically, when you reduce, you know, if you reduce the inhibitory cells from 50% to 20%, but increase their strength accordingly, so they're providing the same net inhibition, it makes no difference to the behavior.
But they need to react to the changes
So,
they need to react to the changes in individual excitatory neurons.
no, in small local groups of excitatory neurons, they don't need to, you know, they just need to look at the average local behavior and be responding to that. Yeah.
How do you choose the connection weights, and how finely tuned do they have to be to get these results?
Yeah, so they don't have to be finely tuned, but there are different parameter regimes that you can define. In the 2013 paper, we try to characterize different parameter regimes. So you do have to tune, and you know, you can definitely, it's easy to get instability, but you don't have to fine tune. There's broad ranges. I mean, one thing we show in the 2015 paper is that, I forget which, but you can vary lots of parameters up and down by 10%, and there's no qualitative difference in the behavior. Yeah.
So why don't we make this the last one before we
Okay.
swap up the speakers?
Did that become more difficult? I saw one of the last slides you moved, I guess, from the ring model to the Hinton model.
Right. I mean, basically, we had a 1D model of space with no orientation and a 1D model of orientation with no space. And now we had a 2D model where space was varying across the model and superimposed on that an orientation map.
So when you modified it from the ring, you've got EI pairings that exist throughout here, each with their own stimulus orientation preferences. And as you move to the pinwheel, then you also have connections within the excitatory ones that have preferential connections between them, right? So
Yeah.
I guess I was curious how that transitioned. So because you were talking about the inhibition connection to excitation, and you would think that there has to be some, I think it's like just more complex as you move to that area. And I was curious.
Yeah, so what we've done here is we have, we assume, even in the 2D model, we assume that the E projections, so the projections in the 2D model are a function of the difference in space, Gaussian fall off, times a function of the difference in orientation. That's the probability of connections, and then they're stochastically sampled. We assume that the function of the difference in orientation is the same for the E projection and the I projections. And that's based on, in layers 2 to 4 in the cat from intracellular recording, the excitation received and the inhibition received have the same tuning. If you were to make a rodent model, the inhibitory cells are untuned. And so we could go to an untuned inhibition model, and we haven't done that. I don't think it would change things very much. But we made the spatial fall off much sharper for the I cells than for the E cells in the 2D model.
All right, well,
Okay.
there'll be other chances to talk about more on break or during one of the other breaks, so thank you very much for
Thank you.